Welcome back everybody to the channel that we all love and support, D1 Math. I do really feel the love, thank you so much. Second, today we're gonna unchain the electron transport chain. So we're not gonna dive deep into the chemistry, however, I'm just gonna tell you exactly what you need. The electron transport chain is a common topic in pre-med, med school, and basically anyone in the medical field. So let's dive right in. So, before we start, I just wanted to remind you of a few things. First, that every hydrogen atom has a negative electron and a positive proton. Second, that losing this electron equals oxidation, and gaining this electron equals reduction. Third, this is the structure of the mitochondria. Here we have the outer membrane, and this is the inner membrane. And between them is the intermembranous space, and this innermost part is the matrix. So we can draw it like this. This part is the inner membrane, and here is the intermembranous space, and above here is the matrix. Now let's get to business. What is the electron transport chain? It is electron flow between electron donors and acceptors through protein complexes. As we said, electron donors equal oxidation, and the ultimate donors are NADH and FADH2. Remember, we were talking about NADH and FADH2 as they produce energy, and this is how? Through the electron transport chain. Moving on, electron acceptors equal reduction. And the ultimate acceptor here is oxygen. And this is why we need to breathe every, I don't know how, I don't know, we breathe a lot. So it's a chain of reduction and oxidation reactions, hence the name redox reactions. We can call it that. Now we have 5 protein complexes embedded into the inner membrane. And there are 2 ways to enter the electron transport chain, either through complex 1 or complex 2. So here, complex 1 takes the hydrogen from NADH, so the name is NADH dehydrogenase. So the other name of complex 1 is NADH dehydrogenase. And as we said, this hydrogen atoms have 2 parts, negative electron and positive proton. So we're gonna pass this negative electron to the next part of the chain, which is coenzyme Q or ubiquinone. And we just throw this positive proton into the intermembranous space. Keep in mind that the inner membrane of the mitochondria is impermeable to protons. The other way that we can enter the chain is through complex 2. Then our donor here is not NADH, but FADH2. And the complex name is succinate dehydrogenase. Remember from the Krebs cycle enzyme, it's the same enzyme. And unlike complex 1, there is no proton to throw into the intermembranous space. And this is why FADH2 produces less energy than NADH. And again, we're gonna pass this negative electron to the next member of the chain, which is coenzyme. So complex 1 and complex 2 just throw electrons to coenzyme Q. Then coenzyme Q is gonna pass the electron to complex 3, which throws protons into the intermembranous space. Then we pass the electron to cytochrome C, which passes it to complex 4, which throws some protons in as usual. So basically all of the complexes throw protons into the intermembranous space, except complex 2. Here this complex 4 does not pass the electron to complex 5. Instead, it gives it to its final acceptor, which is oxygen. Since complex 4 deals with oxygen, just like the red blood cells, it can have either ferrous or ferric ion, depending on if it's oxidized or reduced. As you can see here, we have a lot of protons accumulated on one side of the membrane. And this is called the proton gradient. And this is because, as we said earlier, the inner membrane is impermeable to those protons. So this proton gradient is very important because it's gonna create electrochemical gradient across the membrane. As you can see here, it's too much poor protons wanting to go to the other side inside of the matrix. It's just cruel to not let them in, right? This is why we here have our hero complex 5 or ATP synthase. He's gonna let the poor protons into the matrix. However, he's gonna use their energy to phosphorylate ADP and create ATP, and hence the name ATP synthase. It's the enzyme that makes ATP. Now on this side we have oxygen plus electron plus proton. And as we said earlier, an electron and a proton give you hydrogen atom. So if we have oxygen and hydrogen, then we're just gonna have water, H2O. Now let's apply this knowledge in a pharmacology/physiology manner. Let's have a general rule. Anything that would inhibit any of the 5 complexes will inhibit the electron flow and also inhibit the ATP production. So, electron transport chain complex inhibitors such as complex 1, complex 3, and complex 4 will decrease electron transport chain and decrease ATP synthesis and decrease proton gradient because the complexes are not throwing in the protons. So what about complex 5 inhibitors? It's basically the same as we just said, but with just a tiny bit of change. It increases the proton gradient because you're not letting the poor protons into the matrix, as we said. So complex 5 or ATP synthase inhibitors will decrease electron transport chain flow and decrease ATP synthesis but increase the proton gradient. The last thing is called uncoupling agents. And these agents do not actually inhibit any of the complexes. Therefore, the electron transport chain flow is intact. It's not affected. However, they increase the membrane permeability to protons, meaning they're letting the protons pass into the matrix but not through the ATP synthase. And as you can see, if we do that, we're gonna lose our proton gradient and lose the ability to produce ATP. Since we're not using this energy created by the electron transport chain to produce ATP, then this energy is just gonna go wasted in a form of heat. So they can cause hyperthermia, meaning the body temperature goes very high. So let's go through some examples of the electron transport chain inhibitors. Starting with complex 1, we have two things, rotenone and barbiturates. Complex 3 inhibitor is antimycin A, and complex 4 inhibitors, again, we have two things. First, carbon monoxide, CO. As we said earlier, iron is part of complex 4. So if it's in the first state, it binds to CO. If it's in the ferric state, it binds to cyanide. So cyanide and CO are the inhibitors of complex 4. Moving on to complex 5 or ATP synthase. The inhibitor is oligomycin. And we have 3 examples of the uncoupling agents. First, aspirin. Second, DNP, dinitrophenol, and third is thermogenin. And that, my friends, concludes our quick recap of the electron transport chain. Please let me know in the comments your feedback, and also please let me know which topic do you want us to discuss in the next episode. I will reply to every individual comment And if you have reached this part of the video, thank you so much for your support, and I'll see you in the next one.